Hello everyone, a very welcome you all to Vyas Edification Kota, and today in the series of NCERT discussion we are going to discuss a new chapter from class 12, that is Principles of Inheritance and Variation. Alright, so your NCERT of this chapter actually contains total 16 questions in it, and we are going to discuss each and every question here in detail. So the very first question of this chapter is, mention the advantages of selecting P plants for experiment by Mendel. So we have to mention some advantages why the scientist Mendel sir has selected P plant for his experiment. So pea plant have various kind of advantages. That's why Mendel sir has selected this plant. So first of all, if we discuss the advantages of pea plant, the first advantage of it is it have various contrasting traits. It have various contrasting characters which are easy to know, and they are passing. Which are easy to know, and they are passing from one generation to next generation. When they are passing from one generation to another generation, it's easy to know. It's easy to mention that these characters are passing from parent generation to offsprings or progeny. Alright, so it's having various contrasting characters. The first is this advantage. Second is the pea plant have bisexual flowers. Pea plants have bisexual flowers which undergo self-pollination. So this is also an advantage. So, in case if any insect is not available to pollinate the plant, then they undergo self-pollination. This is also an advantage. Next advantage, if we talk about the pea plant, it's easy to practice cross-pollination in the flowers of pea plant. It's easy to practice cross-pollination in pea plant. Alright. Next, if we talk about their life cycle, their lifespan, their life cycle is actually smaller. So it's easy to experiment, it's easy to work on pea plants or any plant which have short life cycle. So this is one of the advantage, that pea plants are having short life cycle. Alright. Now, if we talk about another advantages, then it produces a large number of offsprings. It produces large number of offsprings in one generation, and it's easy to grow. All these are the advantages why Mendel sir has selected pea plants. And if we write the answer of question number 1, then the advantages of selecting pea plant are as follows. Now we are going to write all the advantages below here. So first one is, it have various contrasting characters. It have various contrasting characters such as seed color, seed shape, such as seed color, seed shape, plant height, and flower. All these are the different characters which are easily available as their contrasting traits in the plant of pea. The next advantage, if we talk about, is this plant bears bisexual flowers. That means the flower which is containing both the reproductive organs such as male and female. So this pea plant having bisexual flowers, and hence it can undergo self pollination in case in case if there is no pollinating agents such as insects are available to them, then they can undergo self-pollination because they are having bisexual flowers. It is also easy. It is also easy to practice cross-pollination in it. Cross-pollination can be done easily in these plants or in the flower of a pea plant with the help of artificial hybridization. Alright, so we can practice easily the cross-pollination thing in the flowers of pea plant. The next advantage of pea plant we talk about is their life cycle. Their life cycle is short, hence they are easy. Their experiment which we are working on it is actually easy. Then next is many offsprings are produced. Many offsprings are produced at a time in one generation. So this is also an advantage that in one generation only it can produce a large number of progeny. And the last but not least, that it is easy to grow. These are the actually advantages of pea plants, why Mendel sir has selected this pea plant. And this is also a criteria which actually can be basis of experiment to anyone. These are the criteria, or these are the thing which can any scientist think about, that these criteria, if any plant or if any sample is actually filling all these criteria, then the experiment would be easy, comparatively easy. So all these are the advantages, and because of these advantages only, Mendel has selected for his experiment as Pisum sativum or pea plant. So this is the complete solution of question number 1. Now moving on to the question number 2, and here we have to differentiate between these two terms. A we have given as dominance and recessive. So we have to differentiate between dominance and recessive. If we talk about the difference between these two, then the dominant allele is able to express itself even in the presence of recessive allele. We have two types of alleles, alright, the dominant allele and recessive allele. So in case of dominance, the dominant allele can or is able to express itself in the presence of recessive allele. Whereas if we talk about recessive one, then it is unable to express itself in the presence of dominant allele. If we talk about another thing, the dominance, then dominant allele will not require, it does not require the similar allele to express its expression. Whereas if we talk about in the recessive one, it requires a similar allele. It requires a similar allele to express itself in the phenotype. Alright, in the homozygous recessive condition only, the recessive allele will express itself in its phenotype. Whereas in dominance, if we talk about, the dominant allele will not require the similar allele to express itself in the phenotype. If the dominant allele is present with recessive one, then it is also able to express itself in the phenotype. Or even if the dominant allele is present with it, then it is also able to express itself in the phenotype. So this is no matter that the dominant allele will require the similar allele to express itself in its phenotype. The next thing, if we talk about these two, then the dominant allele will produce functional enzyme to express itself in the phenotype, whereas recessive allele produces incomplete or defective polypeptide or enzyme which is not capable of expressing the character completely. So if we talk about the difference between these two, in the left column, we are going to talk about the dominance. Dominance. Here we are going to talk about the recessive. Recessive. Alright. So first difference between these two is it is able to express itself. We are talking about dominant allele. Dominant allele is able to express itself even in the presence of recessive allele. Recessive allele. Whereas if we talk about the expression of recessive allele, then recessive allele is not able to express itself in the presence of dominant allele. Dominant allele. Alright, this dominant allele can express its expression or is able to express itself even in the presence of a recessive allele, whereas this recessive allele is not able to express itself if it is present with the dominant allele. The next thing, if we talk about the difference between these two, is it does not require the similar allele to express in the phenotype. For example, if we take example, so this capital T is actually a allele of tall height in the plant. This dominant allele is able to express itself even in the presence of another dominant allele, even in the presence of another recessive allele. Alright, so if this dominant allele is present with dominant allele, if it is present with recessive allele, it will going to show, or it will going to express its expression in the phenotype, no matter whether it is present with the similar allele or whether it is present with the dissimilar allele. Whereas if we talk about the recessive allele, then it requires the similar allele. It requires the similar allele to express in the phenotype. For example, if we talk about this one, then let's take this. This is the recessive allele for the height of a plant, that means dwarf. If it is present with this capital T, this is not possible. If we writing this capital T, it will be in this position, so automatically this will be tall. So to express its expression, or to express its expression in phenotype, it have to present with another similar allele, and in the similar allele if we talk about, then this case would be recessive, and the recessive case makes the plant dwarf, if you are talking about height as the character. So in the recessive allele, it requires similar allele. It requires similar allele means it requires the another recessive allele to express itself in the phenotype. Next, if we talk about the dominant, it produces the functional enzyme. always it produces the functional enzyme, whereas if we talk about the recessive one, it produces the incomplete or defective enzyme. So the result would be maybe incomplete or codominant. So this is the basic difference between the dominant and recessive allele. Dominant is able to express itself even in the presence of recessive one, and recessive is not at all able to express itself in the presence of dominant allele. This one will not require any similar allele to express itself in any phenotype, and this will require the similar allele. The similar allele, that means the recessive allele, another recessive allele, it will require to express itself in the phenotype. It produces functional enzymes, and whereas it produces dysfunctional or destroyed enzymes or defective or incomplete enzymes. So this is the difference between dominance and recessive. So now let's talk about the B part of second question, that is differentiate between homozygous and heterozygous. So now we have to differentiate between these two terms, that is homozygous and heterozygous. So first, if we talk about homozygous, then homozygous is a pure. It is pure for a trait and breeds true. If we are talking about the homozygous one, then this is a pure and it breeds true. That means it produces the individuals with similar genotype. It produces the individuals with similar genotype which are homozygous in condition. That means capital T or small t, for example. And whereas if we talk about the heterozygous one, then this is seldom pure and produces the offsprings with different genotypes. Heterozygous are seldom true and produces the offsprings with different genotypes such as capital T, small t, capital T, small t on the selfing of a heterozygous pair of alleles, that is capital T, small t. If we talk about the homozygous again, then a pair of allele contains similar allele, a pair of gene, a pair of allele. We know that alleles are always present in pair. So these are two are the similar. These two are similar in homozygous condition, whether it is dominant, whether it is recessive. It can be dominant, it can be recessive, but they're always similar. Both the alleles in case of homozygous condition are similar for a trait, whereas in the heterozygous condition, both the alleles for a trait are dissimilar. That means capital T, small t. It can never be capital T, it can never be small t, whereas this condition applies for homozygous one. That means if alleles of a trait are present in the homozygous condition, it would be small t, capital T, but can never be capital T, small t. Now if we talk about the another thing about homozygous condition or homozygous pair of trait, then they can produce only one type of gamete. These homozygous pair can only produce one type of gamete, whereas the heterozygous can produce two types of gametes. Alright, they can produce two types of gametes. Now if we talk about these, then we will going to write the answer. In the left column, we are going to write homozygous, Here we are going to talk about heterozygous. Now if we talk about the difference between these two, the first basic difference is it is true, it is pure. First of all, it is pure and breeds true, What does that mean? That is, it produces the offsprings, with homozygous condition, which are similar to it homozygous condition. Whereas if we talk about the heterozygous one, then they are seldom pure and produces the offsprings with different genotype, That means capital T, capital T, capital T, capital T, small t, capital T, capital T, small t on the selfing of a heterozygous pair such as capital T, small t. Whereas if it is producing any offspring, it is making any individual, then this individual will be present in a homozygous condition, and we know that homozygous condition can be capital T, small t, small t. It can never be capital T, small t. Whereas if we talk about the heterozygous one, then heterozygous one is capital T, small t always. It can never be capital T, small t. It was always present with two different alleles. Alright, and in this condition, both the alleles are similar. So the second basic difference is both alleles of a trait are similar. Both the alleles for a trait are similar, and in case of heterozygous condition, both alleles are dissimilar. As we can see that both the alleles, this is the allele for height of a plant, and both the alleles of a trait are actually present in the dissimilar condition. The third basic difference between these two is it can produce only one type of gamete always. At the time of gametic formation, it can only produces one type of gamete, whereas this heterozygous condition can produces two types of gametes. Now, it does not show, the last but not least, it does not show extra vigor, whereas this one can show extra vigor, means hybrid vigor, which is known as hybrid vigor. That means it can produce hybrids, alright, and it cannot produce hybrids. So this is the basic difference between homozygous and heterozygous pairs of a trait. Homozygous are always present in the similar condition. Also, there is one more difference between this, than that the homozygous condition can either have dominant alleles or can have recessive alleles. It can have either dominant alleles and it can have recessive alleles. Whereas in the heterozygous condition, it contains both the alleles, both the type of alleles, that is dominant and recessive. Heterozygous condition or heterozygous pair of allele can have both the type of alleles, that is dominant and recessive, whereas homozygous one can either have only dominant alleles or recessive alleles at a time, because it is present in a similar condition and the alleles of a trait in the heterozygous condition are dissimilar. So these are the basic differences between homozygous and heterozygous conditions. Now we are having the third part of question number 2, that is differentiate between monohybrid and dihybrid. So we know that what is monohybrid cross and dihybrid cross. Monohybrid is actually a cross between two pure organisms in order to study single pair of alleles, in order to study the inheritance of single pair of alleles for a trait. Alright, so this is about the monohybrid cross. If we talk about the dihybrid cross, then it is also a cross between two organisms in order to study the inheritance of two different alleles, two different pair of alleles belonging to the different characters, So this is about dihybrid cross. In case of monohybrid, it produces a phenotypic ratio in F2 generation as 3:1. Whereas in dihybrid cross, we see in the F2 generation as phenotypic ratio 9:3:3:1. Whereas if we talk about the genotypic ratio in both of these, then monohybrid cross have 1:2:1 genotypic ratio, whereas the genotypic ratio of dihybrid cross is larger, such as 1:2:1, 2:2, 4:2, 1:2:1. So this is a large one ratio. Now we are going to write the differences between monohybrid and dihybrid over the screen. In the left column, we are going to write about the monohybrid. Both these are the crosses. Dihybrid. So first is, it is a cross. It is a cross between two pure organisms, in order to study, why do we these experiment? To study the inheritance of genes, in order to study the inheritance of alleles for a trait. We want to see how a trait is passing from parent to offspring generation. So monohybrid is a cross concerning with the study of inheritance of only single pair. Inheritance of a single pair of allele. Alright, and if you talk about the dihybrid cross, then it is a cross between two organisms in order to study the inheritance, of course, but of not a single pair but of two different pairs of alleles for a trait. So in order to study the inheritance of two different pair of alleles belonging to different characters, There will be two characters which we are noticing, which are passing from parent generation to next generation or their offsprings into this cross, that means dihybrid cross. That is why the name has given as dihybrid. That means two characters are passing from one generation to next generation. We are noticing the two characters at a time, and in this cross we are just noticing only one single character which are passing from parent to offspring generation. That is why the cross is being named as monohybrid. That means one character is passing from parent to next generation. So this is a monohybrid cross and this is dihybrid cross. If we talk about the ratios, then phenotypic ratio in F2 generation, in case of monohybrid cross, we have 3:1 ratio. Alright, 3:1 ratio, that means 3 are tall and 1 is dwarf. And in case of dihybrid cross, we have 9:3:1 as phenotypic ratio. Whereas if we talk about the genotypic ratio of these two, then the genotypic ratio in F2 generation, it is having 1:2:1. Alright, and this is having 1:2:1, 2:4:2:1:2:1. This is the genotypic ratio in the F2 generation of a dihybrid cross. So this is the basic difference between monohybrid and dihybrid crosses, and this is a complete solution of the part C of question number 2. Now let's come towards the next question, that is question number 3. A diploid organism is heterozygous for 4 loci. How many types of gametes can be produced? The question is asking if any diploid organism is heterozygous for 4 loci, then this particular organism can be capable of producing how many types of gametes? So, if any organism is heterozygous for 4 loci, that means the organism is heterozygous for 4 characters. Alright. And if we talk about the gametes, then the gametes produced based on the number of heterozygous characters in an organism is 2 raised to the power n, wherein the n is the number of heterozygous characters in an organism. So in the given condition, the n will be 4, and if we solve the question, then it will be like if we talk about 2 की power n, 2 raised to power n, then n we have is 4 here. In this question we have 4 as n. So if we want to solve this, then it will be solved like this, and the total number of gametes we will find is 16. Alright, so total number of gametes will be 16. So let's write the answer, that a diploid, this is the formula for obtaining different gametes from a organism. So a diploid organism is heterozygous for 4 loci. That means the organism is heterozygous for 4 characters. Alright. Now if we talk about the gametes produced based on the number of heterozygous characters is 2 raised to the power n. This is the formula to solve how many types of gametes are present in an organism, or how many types of gamete can organism produce. So if we talk about this, then this is a formula to solve this, and whereas n is equal to the number of heterozygous characters. So according to this question, we have 4 loci. An organism, a diploid organism, have 4 loci. So that means an organism, a diploid organism, have 4 heterozygous pairs, heterozygous characters. So we are having n as the number of heterozygous characters. So automatically this 2n will have, as we know that n is heterozygous character. So according to this question, we have n is equal to 4, and if we try to solve this, we will get 16. So 16 is the answer. 16 gametes are going to be formed by this particular organism, which is diploid, and have 4 different loci for heterozygous characters. So now this is the complete solution of question number 3. You can solve the type of gametes produced by an organism by this formula, that is 2 raised to the power n. Now let's talk about question number 4. Explain the law of dominance using monohybrid cross. So we have to explain law of dominance with the help of a monohybrid cross over here. First of all, we are talking about what is law of dominance. So law of dominance states that a dominant allele of a character in a monohybrid cross expresses itself and suppresses the expression of another recessive allele. This is law of dominance. Law of dominance states that a dominant allele expresses itself in a monohybrid cross, whereas it suppresses the expression of another allele, that is recessive allele. For example, if we talk about a monohybrid cross by Mendel sir, he crossed the pure tall-heighted plant with pure dwarf plant. In the F1 generation, what he found that the recessive one expresses itself, the dominant express itself. In the F1 generation, all progeny were tall. There were no progeny, there were no individual with dwarf height. Whereas this dwarf heighted plants or small plants expresses itself in the next generation. So this is what happens in the law of dominance. This states that the dominant allele expresses itself in a monohybrid cross where it suppresses the expression of recessive allele. This recessive allele is not lost, it remains there. It remains hidden or masked in the progenitors of F1 generation, and it reappears in the next generation. This is what happens in the law of dominance, that in F1 generation, the dominant allele will express its expression in the progeny, in the phenotype, and the recessive allele is not lost, it remains masked or it remains hidden in the progeny of F1 generation, but it reappears in the next generation, that means in the F2 generation. When Mendel crossed the heterozygous plants between themselves, when the selfing was done between the heterozygous tall plants, we found that in the F2 generation, some plants were tall, some plants were heterozygous tall, some plants were homozygous tall, and the recessive pair of allele reappeared, which was similar to the one of its parent. So this recessive allele expresses itself in the next generation. It expresses itself in the F2 generation. So this is the law of dominance with the help of a monohybrid cross, and here we are going to write the answer for, we are going to write the law of dominance, and we will also make a monohybrid cross to explain the law of dominance. So first of all, law of dominance states, that the dominant allele express itself in a monohybrid cross, and suppresses the expression of a recessive allele. It expresses itself. The expression we are getting in a monohybrid cross is of dominant allele, of course, and the recessive allele is that does not mean that it is lost. It is remain there. It is remain hidden there. It is remain masked there in the progeny of a F1 generation. So this is law of dominance, and this recessive allele is not lost. It is not lost. However, we are not able to find this recessive allele in the F1 generation or in the progeny of F1 generation, but it will reappear in the next generation. It will reappear in the F2 generation. So, law of dominance states that a dominant allele expresses itself in a monohybrid cross and it suppresses the expression of a recessive allele. However, this recessive allele is not lost. It is remain masked or hidden. It is hidden or masked in the progenies of F1 generation. Alright, now we are going to make a monohybrid cross to explain this law of dominance. We know that law of dominance is actually where or when the dominant allele expresses itself and suppresses the expression of recessive allele. This recessive allele is going to express in the next generation or F2 generation. So let's talk about that. We know that Mendel crossed homozygous tall-heighted plant with dwarf homozygous plant. This is homozygous pure tall plant, and this is pure dwarf plant with homozygous condition as small t. So now I am writing here it as dwarf because this is representing dwarf plant and this is representing tall plant. What he did, he actually crossed these two plants. And in the F1 generation, what he found? In the F1 generation or in the first progeny, what he found that all plants were tall. But are heterozygous tall. He found that in F1 generation, the phenotype or the appearance of all the plants were tall, but genotypically they all are heterozygous tall. They are not similar to their parents. See, these progeny or these individuals in the F1 generation are not similar to one of these parents. These are not similar to this parent and these are not similar to this parent as well. Alright, so in the F1 generation, what he observed? He observed that on crossing this pure tall-heighted plant with this pure dwarf plant, he got all the progeny as tall plant but in heterozygous condition. Then where is this recessive allele? Where is this recessive allele? If it is not expressed in the F1 generation, it may be lost, but this is not true. It is not lost. It remains hidden in the progeny of this generation 1. Here you can see this recessive allele, it is containing the recessive allele, it is also containing the recessive allele. All the plants are containing the recessive allele, but it cannot express itself because it is present with dominant one. And this is the rule that recessive allele cannot express itself in the presence of a dominant allele. It is only capable of expressing its expression in the presence of similar allele. That means a recessive allele. If the recessive allele is present with recessive allele, another recessive allele, then only the expression will come out. So what he done? He took one plant and he self-fertilized it. Alright. So what he done? Self-fertilized it. The plants of F1 generation self-fertilized with each other. So what he observed in F2 generation, he observed, first of all, These are going to make gametes, and the gametes we will get like this. The gametes we are going to make Punnett square over here. This is Punnett square. And what he observed in F2 generation, when he self-fertilized these plants, he self-fertilized the plants of F1 generation, then he found that a tall plant, pure homozygous tall plant, heterozygous tall plant, again a heterozygous tall plant, and a dwarf plant expressed in the F2 generation. That means the recessive allele is not lost, the recessive allele was not lost, they were remain hidden in the progeny of F1 generation and expressed, reappeared in the F2 generation, in the progeny of F2 generation. Here you can see when he self-fertilized the progeny of F1 generation, he observed that the maximum plants were tall, some of them were heterozygous or tall, some of them were homozygous tall, and some of them are dwarf, which are exactly similar to one of its parent generation. Alright, this is actually similar to one of its parent, which is reappeared in the F2 generation, which was not seen in the F1 generation. So this is the law of dominance with the help of a monohybrid cross. You have to remember that in the law of dominance, the recessive allele, which is not able to express itself in the F1 generation, is not lost at all. It remains hidden, it remains mapped or masked in the progeny of F1 generation, but it will reappear, it will express itself in the next generation, that is F2 generation. So this is the complete solution of question number 4. Now let's talk about the question number 5, define and design a test cross. So first we have to define a test cross, then we will going to design test cross. So test cross is just an experiment in which organism showing dominance for a trait has to be tested for its genotype. This is the basic definition of a test cross, that test cross is an experiment. Test cross is an experiment showing a dominance, in which organism is showing dominance for a trait has to be tested for its genotype. To test the genotype of an organism, we use recessive allele, recessive pair of alleles. Alright. Now, when an organism is showing dominance for a trait, it could be homozygous in condition and it could be heterozygous in condition. Now when we experiment, when we do experiment to examine the genotype of an organism, we know that we use recessive pair of allele, recessive parent to test the genotype of an organism. If the organism is homozygous in condition and we do experiment as test cross, then the offsprings, all the offspring are same. And if the heterozygous condition, if the genotype of an organism is heterozygous, then half of the offsprings will be homozygous and half of the offspring will be heterozygous. So let's talk about this question number 5. Test cross is an experiment. Test cross is an experiment in which the organisms showing dominance for a trait has to be tested for its genotype, whether the genotype is heterozygous or whether the organism is heterozygous or homozygous. This we have to test, and for this test we use homozygous recessive parent. So test cross is just an experiment in which the organism showing dominance, for a particular trait has to be tested for its genotype, whether the genotype is heterozygous, whether the genotype is homozygous. What is the genotype of that organism? To test the genotype of that organism, we use this test cross as an experiment. And to test the genotype of an organism, we use recessive parent. Alright, so we use recessive parent. We use recessive parent to test the genotype of an organism. When any organism which is being tested, for its genotype and showing dominance, it could be homozygous, or it could be heterozygous, Now let's talk about the test cross. We'll be going to design a test cross over here. As we know that to test the genotype of any organism, we will use recessive parent. We will use homozygous recessive parent. So let's talk about the height of a plant. If any organism is homozygous tall, let's talk about the first case. We are going to discuss two cases. This is first case in which the organism is having, suppose the organism is having capital T condition, that means homozygous dominant condition, but we don't know what is the genotype of that organism. We have to still test the genotype of this organism. Now to test the genotype of this organism, we will mate with this organism is small t. We are going to mate this organism with recessive parent. This is the recessive parent we know. This is the homozygous recessive parent, and in a test cross, which is an experiment to determine the genotype of an organism, always the homozygous recessive parent is being used. Now in case number 1, if any organism is homozygous dominant, then what we do in a test cross, we mate this organism with recessive parent. Now what is going to happen? It will produce gamete as capital T, it will produce gamete as small t. All the offsprings without exception in F1 generation will be tall. All the offsprings in F1 generation will be tall. That means heterozygous, tall. All the offsprings will be heterozygous. So this is the conclusion, this is the result. We don't know the genotype of the parent of an organism or an organism. We are just testing the genotype of that organism, and for testing we are going to mate this organism with recessive parent. Now what happens? These two are going to make gametes. This has made capital T as gamete and this has made small t as gamete. When they fuses with each other, they produces offsprings of F1 generation. All the offsprings in F1 generation are heterozygous tall. All the offsprings are heterozygous. If in the conclusion of a test cross in the F1 generation we found the offsprings, all the offsprings are heterozygous in F1 generation, then we can say that the organism, by this conclusion we can say that the organism, what organism we are talking about? This organism. The organism is homozygous. The organism is homozygous. Alright. So by this conclusion we can say that the organism is homozygous in condition. Now let's talk about the second case. Suppose We don't know the genotype of the organism. Suppose we are taking the heterozygous condition here. We don't know the exact genotype. We will conclude a result from the F1 generation of it. Let's talk about here the next case, that is heterozygous condition. We will make mate this with recessive parent again. We will make this possible. We will mate this organism with recessive parent. This will produce two types of gamete, as this is heterozygous in nature, and this is going to produce only one type of gamete. Alright, now what happens in F1 generation? Half of the generation, half of the progeny in F1 generation will be heterozygous, and half of the progeny in F1 generation will be homozygous in condition. Alright, so what we will get by the conclusion from F1 generation in second case, that is half of the progeny is homozygous, half of the progeny is homozygous, and half of it is heterozygous. Alright, half of it is heterozygous. So we can conclude now, by making these organisms mating with recessive parent, we can conclude the result. If all the organism in F1 generation are heterozygous in nature, then we can say the organism is having homozygous condition, homozygous genotype. And if all the progeny, we observe the progeny in F1 generation, all the progeny in F1 generation, if found to be half are the heterozygous and half are the homozygous, then we can conclude that this organism is actually heterozygous in nature. So this is the complete solution of question number 5. So now let's talk about question number 6. Using a Punnett square, work out the distribution of phenotypic features in the first filial generation after a cross between a homozygous female and a heterozygous male for a single locus. So now what we have to do in this question number 6, we have to make a Punnett square. We have to make making a female and male cross with each other for a single character only, and we have instructed that we have to take a homozygous female, we have to mate this homozygous female with the heterozygous male. So we have to make a Punnett square, and we have to find the phenotypic ratio. We have to make a Punnett square to find phenotypic ratio in the first filial generation or F1 generation. We have to find phenotypic ratio in F1 generation or first filial generation after a cross between a homozygous female and a heterozygous male for a single locus or character. Single locus means single character we are talking about. So now what we are going to talk about, then we will take parent, we will mate the parents, the given parents. So as we are instructed to take heterozygous male for a single locus or a single character, the character we are choosing here is height. Alright, so the character we are choosing as height. So heterozygous male will be capital T, small t. This is a male with heterozygous condition, is mating with homozygous female, That's what we are instructed to do. So we are taking heterozygous male and the character we are choosing is height. So heterozygous male we have taken and homozygous female we have taken. These are going to make gametes, as we know that this heterozygous pair is going to make two types of gametes, capital T and small t. These are gametes. And this female is going to make only one type of gamete, that is small t. Alright, now we are going to make a Punnett square over here. We are going to fuse these gametes with each other. So now On this, we are going to write the gametes of a male, capital T small t. Capital T small t are the gametes, capital T small t. And now we know that female is going to produce only one type of gamete. Then we are going to write the gametes over here of a female. Now we are crossing them. We are matching them. So capital T small t we are obtaining here. Here we are obtaining small t, small t. Here we are obtaining capital T, small t, and here also we are obtaining small t, small t. So you can see this is the F1 generation or first filial generation, F1 generation or first filial generation, and according to the question we have to find the phenotypic ratio in F1 generation only. So from this Punnett square we can conclude that the phenotypic ratio, we are not talking about the genotypic ratio, the phenotypic ratio in F1 generation we are obtaining as 1:1. So 1:1 is the phenotypic ratio of F1 generation. Alright, so in F1 generation 1:1, that means 50% of the individuals, 50% of the individuals are dwarf or 50% individuals are heterozygous tall, or let's leave heterozygous. What is conclusion? That 50% of the individuals of F1 generation are small and 50% of the generation, 50% of the F1 generation are tall in nature. So this is the complete solution of question number 6. Let's talk about question number 7. When a cross is made, between tall plant with yellow seed, that is capital T small t, capital Y small y, and tall plant with green seed, capital T small t, capital Y small y, what proportions of the phenotype in the offsprings could be expected to be tall and green, dwarf and green? So to obtain these results, to examine this, what proportion on the phenotype of a plant is actually having tall and green and dwarf and green. So we have to make a cross. First of all, we will select parents, which we are already provided with. So this is the one parent, capital T small t, capital Y small y, that is tall plant with yellow seeds. Now is being crossed with another parent, which is tall plant with green seed. So capital T small y small y. Now they are going to make gametes. Different type of gametes we will obtain from here. Gametes are capital T, capital Y, capital T small y, small t, capital Y, and small t small y. These are the different types of gametes obtained from this parent, and this parent over here is going to make only two types of gametes, that is capital T small y and small t small y. Now we are going to make a Punnett square over here with these gametes, and we are going to fuse these gametes with each other to obtain a result. Here we are going to make or write the gametes. So first gamete we are taking the gametes capital T small y, small t small y. Here we are writing these gametes capital T capital Y, capital T small y, small t capital Y, and the last is small t small y. Now we are going to fuse them with each other. So the first result is getting capital T, capital Y, small y. So this is a plant with tall height and yellow seed. This is a plant with tall height and yellow seed. Next is capital T, small t, capital Y, small y. This is also a tall plant with yellow seed. Second is capital T, small y, small y. This is a plant with tall height but with green seeds. Next is capital T, small y, small y. This is also a plant with tall height and green seeds. Now next is capital T, capital Y, small y. This is also a plant with tall height and yellow seed. Then small t, capital Y, small y. So this is a plant with dwarf height and yellow seed. This is a dwarf plant with yellow seed. Then we talk about the next result, that is capital T, small y, small y. This is a tall plant with green seed. Then small t, small y, dwarf and green seeds, dwarf plant with green seeds. So now we have to conclude what proportion of the phenotype in the offspring is tall and green and dwarf and green. So first, if we talk about tall and green means the height of a plant should be tall and the seed are green, and the seed we will obtain will be green and in recessive condition only, that means small y. So this is not the condition applying to this. We have to find among all these offsprings, we have to find tall and green. This is tall and yellow. This is tall and yellow. This is tall and green. So this is tall and green 1. Then this is also we have tall and green plus 1. Then this is we have tall and yellow. This is we have dwarf and yellow. This is we have tall and green. This is we have tall and green. So plus 1. This is we have tall and green. And the last one we have is dwarf and green. So these are the three. Three are the tall and green. Three are the tall and green among all these offsprings. Similarly, we have to find among all these offsprings what proportion of the phenotype of the offspring is having dwarf and green. Dwarf and green is only one. This one is only dwarf and green. Dwarf and green. So only one phenotype is showing dwarf and green among the offsprings of this cross. So this is the complete solution of question number 7. Let's talk about question number 8. Two heterozygous parents are crossed. If the two loci are linked, what would be the distribution of phenotypic features in F1 generation for a dihybrid cross? So this question is basically asking about linked genes. Now what is linked genes? Linked genes is described or is defined as the presence or coexistence of two or more genes in the same chromosome. Linked genes is defined as the coexistence of two or more genes in the same chromosome if the genes are present on the same chromosome and lies close to each other, then these genes are transferred or inherited from parent generation to their offsprings together. Alright, what is linked gene? Linked gene is defined as the coexistence of two or more genes in the same chromosome. If these genes are present on the same chromosome and lies close to each other, then they are going to be inherit from parent generation to offsprings together. These genes or such genes which are going to inherit from generation to generation together, these kind of genes are known as linked genes. For example, we have example for this. For example, the yellow-bodied, white-eyed and wild-type parent in Drosophila produces a wild-type parent and yellow-bodied and white-eyed parent. The genes in Drosophila, the yellow-bodied gene and the white-eyed gene in Drosophila are linked genes. They are present on the same chromosome and they lie close to each other. Hence, it will transfer into the next generation together and hence they are called as linked genes. So, if we write the answer of question number 8, it would be like this. First of all, we will be writing what is linked genes. So, linked genes is defined as, linked genes is defined as the coexistence, of two or more genes in the same chromosome. In the same chromosome. If the genes if these genes are present on the same chromosome, and lies close to each other, then they are inherit together to the progeny, because they are the linked genes and they are linked together very well. They are lying together, they are lying closely together on the same chromosome. So when the genes are linked, they are inherit together into the progeny or into the next generation. For example, we have example for this. For example, we have example for this as Drosophila. So yellow bodied and white-eyed and wild type of parent, wild parent in Drosophila, produces yellow bodied white-eyed and wild type of offsprings. Alright, now what happens, this yellow bodied and white-eyed and wild type of parent in Drosophila produces yellow bodied and white-eyed wild type of offsprings. The sex are interchanged. Alright, the sex are interchanged and the linked genes are interchanged. Alright, why these linked genes of yellow body and white eyes are changed? Because there is linkage. So linkage is seen in the genes of yellow bodied and white eyed. This is because the genes of yellow bodied and white eyed are linked, and because they are linked, they will be inherit together into the progeny. Alright, they are linked together and hence they are inherited together into the progeny or in the next generation. So this is the complete solution of question number 8. Now let's talk about question number 9. And this question is asking, briefly mention the contribution of T.H. Morgan in genetics. So basically in the study of genetics, we study Mendelism. The first part of genetics is about Mendelism, that is a scientist Mendel, and the second part of genetics is about T.H. Morgan. This scientist T.H. Morgan has contributed in the genetics very much, and the topic linkage and recombination is from this scientist only. So first of all, if we talk about the contribution of T.H. Morgan, what he has contributed in the genetics, as I mentioned already, that he contributed or he proposed the concept of linkage and recombination. Basically the concept of linkage actually introduced by another scientist such as Sutton and Boveri, but the experimental proof was given by this scientist named as T.H. Morgan. Now as we know that Mendel performed many experiments in genetics. On which plant? Pea plant, Pisum sativum. He chose the Pisum sativum for his experiments. Whereas if you talk about this scientist, T.H. Morgan, he chose Drosophila. Drosophila, which is a fruit fly. Drosophila melanogaster is the complete scientific name for fruit fly. He chose Drosophila melanogaster, named such as fruit fly, for his experiments. Alright. So, T.H. Morgan introduced linkage and recombination. T.H. Morgan described linkage as the presence of or coexistence of two or more genes in the same chromosome, which are lying on the same chromosome and lies close to each other, which are going to inherit together into the progeny. These genes are named as linked genes by T.H. Morgan. He also performed dihybrid crosses in Drosophila to show that linked genes are inherited together into the progeny and are present on X chromosome. By the dihybrid cross performed in the Drosophila melanogaster by the scientist T.H. Morgan showed us that linked genes are inherited from generation to generation together and are present on the sex chromosome, that is X chromosome. Alright. Also he described that the loosely linked genes show high percentage of recombination, whereas the tightly linked genes on the chromosome show very low percentage of recombination. So this is the contribution which is given by this scientist T.H. Morgan into the genetics. So now let's write the answer of question number 9. First of all, we will write T.H. Morgan worked on the fruit fly such as Drosophila melanogaster. Drosophila melanogaster is the scientific name for fruit fly, melanogaster. Drosophila melanogaster, to show his results, he chose Drosophila melanogaster, a fruit fly, for his experiments. He introduced the concept of, actually he does not introduced, he proved the concept of linkage. He proved the concept of, linkage and recombination. All right. He also performed. How does he actually prove the result of linkage? He performed the dihybrid cross. And because of this dihybrid cross done on the Drosophila melanogaster proved about the linked genes, about linkage. He performed the dihybrid cross on Drosophila to show that the linked genes inherit together into the progeny or into the next generation, into the progeny or next generation and are present. He also proved that the linked genes are present on the X chromosome. So this was the result given by the scientist T.H. Morgan. He also described first of all, he described the coexistence of two or more genes on the same chromosome, lying close to each other, are linked genes, and because they are linked genes, they will be inherit into the progeny together. And also he said that the tightly linked genes show low recombination, while the loosely linked genes show higher percentage of recombination. So this is the contribution of T.H. Morgan into the genetics, and this is the complete solution of question number 9. Now let's talk about question number 10. What is pedigree analysis? Suggest how such an analysis can be useful. So first we have to understand what is pedigree analysis, then we have to mention some uses of it. So first of all, what is pedigree analysis? Pedigree analysis is a chart. It is a chart which shows genetic connection among individuals. Pedigree analysis is just a graphical chart made by doctors, and this chart shows genetic connections among individuals. An analysis of inheritance of traits in a family is known as pedigree analysis. When we analyze the inheritance of a particular character into a family over several generations, this analysis of a particular trait, this analysis of inheritance of particular trait over several generations in a family is known as pedigree analysis. If we mention the uses of it, then it helps, this pedigree analysis helps to identify the origin of a trait and also let us know the flow of that particular trait in a family. This is the one use. Second thing is, it helps to avoid harmful effects of marriages among close relatives. This pedigree analysis helps us to avoid the harmful effects by avoiding marriages into close relations. Alright, this pedigree analysis also helps to identify the expressive or recessive allele in a family which can cause genetic disorders such as color blindness. Alright, so these are the uses. So let's write the answer of question number 10. First of all, what is pedigree analysis? A chart which shows the genetic combination among individuals. analysis of inheritance, an analysis of inheritance of a trait, an analysis of inheritance of a trait over several generations, in a family, over a several generation in a family, over several generations in a family is pedigree analysis, So this is the definition of pedigree analysis. I hope you understood that what is pedigree analysis. It is a chart which helps us to know the genetic connection between individuals or among individuals. When we analyze a particular trait over several generations in a family, that study of that particular trait or inheritance of that trait in a family over several generations is actually known as pedigree analysis, and it is done by doctors. Now if you want to mention some uses of it, uses of pedigree analysis, uses of pedigree analysis. Then the first use if we talk about is, it helps us to know the origin, that means where actually that particular trait is originated in the family. This pedigree analysis helps us to know the origin and also flow, origin and flow of the particular trait in a family. By this pedigree chart, doctor can get us know where the particular trait is originated in a family, and he also can know the flow of that particular trait in a family over generations. Second is, it helps to identify the expressive or recessive allele which may cause genetic disorder. Third use of it we can talk about is, it helps us to avoid harmful effects of marriages among close relatives. Alright, and the last but not least, it also helps in genetic counseling, to know the genetic disorder in children's. By this pedigree analysis, one can get to know the defective allele, one can get to know the defective trait which can cause genetic disorder in children's. By this pedigree analysis, we can get help to know whether the trait is originated, where the trait is originated, in which individual, on which level the trait is originated, if it is defective, or which trait is causing disease or disorder in the family. Alright, we can get to know the origin of that particular trait and also the flow of that trait over several generations into the family. By this pedigree analysis, we also can identify the expressive or recessive allele which can cause genetic disorders in a family or in any individual, such as, for example, color blindness. Alright, so these diseases, these disorders can be identified with the help of pedigree analysis done by doctors. It also helps us to avoid harmful effects of marriages among close relatives. And last but not least, it also helps in the genetic counseling of family to know the genetic disorder in children. And I hope you all get the answer. What is pedigree analysis? It is just a chart which is helping us to know the genetic connection among individuals and the analysis of a particular trait or the analysis of inheritance of a particular trait over several generations in a family is important. In a family, when we doing this pedigree analysis, when we doing or analyzing a particular character, when we analyzing the inheritance of a character over several generations in a family, that particular study of this inheritance of a character is known as pedigree analysis with the help of a chart. It is done with the help of a chart. So this is a complete solution of question number 10. Now let's talk about next question, question number 11. How is sex determined in human beings? How is sex determined in human beings? So first of all, human beings exhibit male heterogamy. Human beings exhibit male heterogamy means a human male can produce two different types of gametes. The gamete containing X chromosome and the gamete containing Y chromosome. Whereas if we talk about the female, the human female can produce only one type of gamete containing only X chromosome. All the ovum or all the eggs are having X chromosome in it. But there are equal chances of sperms in the human male that some of the sperm can have X chromosome in it and some of the sperm can have Y chromosome in it. So on fertilization, if we talk about the sex determination in human beings, how it is done, we know that male heterogamy is shown by the human male and homogamy is shown by the human female. So when a sperm containing X chromosome, fertilizes with the ovum, then the baby will produce as female. And when a sperm containing Y chromosome fuses with the egg, then the baby will produce as male. Hence, the determination of sex is done by the genetic makeup of sperm, and there is an equal chance of a fertilizing sperm being an X or Y. So this is a clear by chance factor that which sperm is going to fertilize the egg. Now we are going to write question answer 11, that humans, it will be start like this, that human beings show male heterogamy, whereas the female homogamy. So human beings show or exhibit male heterogamy and the female homogamy. That means the males can produce two types of gametes containing either X chromosome or Y chromosome. Whereas if we talk about the female case, female will produce only one type of gamete containing one type of X chromosome, this is. Now, when a sperm containing X chromosome, fuses with ovum, the baby produced will be female, because X chromosome of sperm will fuses with the X chromosome of ovum and it produces female child. Whereas if we talk about a sperm containing Y chromosome, when fuses with ovum, it will produces male child. Or male baby. Why is it so? Because the X chromosome of sperm, Y chromosome of sperm fuses with the X chromosome of ovum, because a ovum will always have X chromosome. So the Y chromosome of sperm will fuses when with the X chromosome of ovum, it will produces always male child. Alright, the male child. So this is a by chance factor. This is a matter of chance. And it depends on the genetic makeup of a sperm, whether the baby produced will be female or male. If we make a cross of it, let's suppose if we make a cross of it, then let's suppose this is male and this is female. Male will produce two types of gametes containing X chromosome and Y chromosome, and female will produce only one type of gamete containing only X chromosome. If this sperm containing X chromosome fuses with X, then XX will form a zygote, will contain XX chromosome, 2X chromosome. Hence, we can conclude that the baby produced will be daughter or female. If the Y chromosome fuses with X chromosome of an ovum, then a zygote will contain X and Y chromosome in it, and the zygote, when converted into a new individual, it will form a male child. Whereas this zygote will going to form female child. So this is how sex is determined in case of human beings, and it depends on the genetic makeup of a sperm. So this is the complete solution of question number 11. Now let's talk about the next question, question number 12. A child has blood group O. A child blood group is O. If the father has blood group A, the blood group of father is A and the mother blood group is B. The mother is having blood group B. A child is having blood group O. A father of this child is having blood group A and the mother of this child is having blood group B. Work out the genotypes of the parents and the possible genotypes of other offspring. So we have to know, we have to mention the possible genotypes of their blood groups of the other offsprings. So first of all, this is the case of multiple allelism. Now what is multiple allelism? When one character basically is controlled by two or more alleles. then this character or this phenomena is known as multiple allelism. When any character is controlled by two or more alleles, that phenomena is known as multiple allelism. Similarly, if we talk about the blood group system in human beings, the blood group system, the blood group, which is a character in case of human beings, is controlled by three alleles. We have blood group system, ABO blood group system in human beings is controlled by three alleles, IA, IB and II. We have three alleles in our body. Not in our body, if we talk about individually, any individual or any human being, either it may be male or female, individual will carry only two alleles, because only two pair of alleles are going to control any character. But when we talk about the population, there are three alleles present in a population which is responsible for controlling the character of blood group in human beings. So in a population we have three alleles which are able to control the blood group system in human beings, but in individual only two alleles is controlling the blood group. So in a population, if we talk about in a population of human beings, the blood group is controlled by three alleles, and these three alleles are IA, IB and II. So these two alleles, IA and IB, are dominant. They are dominant, and this II is recessive allele. This II is recessive allele and is going to make O blood group in any individual. So in a population of human beings, the blood group is controlled by three alleles, that is IA, IB and II, among which IA and IB are dominant and II is recessive. But if we talk about on the level of individual or any male or female human, then the individual will contain only two alleles which are controlling the blood group system in it. Alright, now let's move again towards on the question. A child, if a child is having blood group O, then it must have received allele I from one of the parent and also another allele II from another parent. The blood group O will only express itself in the condition when any individual or offspring is having or receiving two alleles as II and II from both of the parents. If both parents are giving or inheriting the II allele into their offsprings for their blood group, then the blood group of that offspring or progeny will be blood group O, just like this. So what is question is saying that the child is having, suppose this child is having blood group O, and according to the question, the father is having blood group A. The father is having blood group A and the mother is having blood group B. Then how is it possible then their child can have blood group O? Yes, it is possible, because the possible genotypes, if we see the possible genotypes of the blood group system, of the ABO blood group system in human beings, then there are 6 possibilities. There are 6 possibilities of the genotypes, as we have 3 alleles, and the formula for obtaining the possible genotypes of an organism for a particular character is 2n. So according to the formula also we have 6 genotypes, possible genotypes, 6 possible genotypes, and also we can determine simply by writing it IA. We are writing the possibilities of genotypes of blood group system. So IA, this is one possibility. IB, these are the 4 possibilities. The next I, A, I, B, I. These are the 6 possible genotypes of ABO blood group system in human beings. These are the possible 6 genotypes. Now, this IA will produce blood group A in any individual. If the alleles in any individual present as IB, if these 2 alleles of blood group system of blood group are present in any individual, IB are present in any individual, then the blood group of that individual will be B. If any individual is having IA and IB alleles in it, then the blood group will be AB. Then the blood group will be AB. If any individual is having allele II in it, then the blood group will be O. Then the blood group will be O, because this is the recessive condition, and in the recessive condition this II allele will going to make O blood group. And in the next case, in this next case, if any individual having allele IA and II, then also the blood group will be A. In the last case, if any individual is having IB and II allele, so the blood group of that individual will be B. Why is it so? Because A is dominant over I. Here also the B is dominant over I. So these are the possible 6 genotypes and their results in the blood group system of human beings. So now we can conclude the child which is having O blood group must be receiving the 2 II alleles from both of their parents. If the blood group of this child is O, then it must have received II alleles from both of the parents, from both of the parents. Then only it is possible to having blood group O in it. Alright, so this is the answer. And if we talk about the genotype of the parents, we have already talked about the genotype of parents. The blood group of father is A. So the father can have this genotype, the father can have IA allele in it, or the father can have this kind of genotype also, IA and II. If we talk about the genotype of mother, as mother is having blood group B, so the mother can have this kind of alleles, IB and IB. Also, there is possibility of having mother IB and II allele, because it is also making a blood group B. So this is the possible genotype of mother and father, and also the possible genotypes of other offspring. These are 6 possible genotypes of other offsprings. Any of the possible genotype can be given or inherited by their parents into their offsprings. So this is the complete solution of question number 12. Now let's talk about question number 13. Explain the following terms with examples: codominance and incomplete dominance. So we have to explain codominance and incomplete dominance along with their examples. So first we are talking about codominance. So if a plant having yellow colored flowers fertilizes with a plant having red colored flowers. If a plant having yellow colored flowers fertilizes with a plant having red colored flowers, then in the F1 generation all the progeny will have orange colored flowers. This is because both the alleles are present in the dominant condition and expressing themselves together. The phenotype is actually a mix. The phenotype is a fix in F1 generation, and this is known as codominance. The example, if we talk about the codominance, the human blood group AB, is an example of codominance. If any individual is having blood group AB, then both the alleles A and B, as both the alleles are dominant, they are expressing themselves together in a hybrid condition. So the blood group in the individual is expressing as AB. This condition is known as codominance. Whereas if we talk about incomplete dominance, then in this condition the genes of an allelomorphic pair are not expressed as dominant or recessive, but they express partially. They express partially. And if we talk about the example of incomplete dominance, we have an example of NCERT as snapdragon. Snapdragon flowers. We know that snapdragon flowers are red color and white colored. There are two plants having red color flowers and white color flowers. But what we found in the F1 generation, all the progeny are having pink color flowers because they are not expressed as dominant or recessive. They are expressing themselves partially. They are expressing themselves partially. The result which is found in the hybrids of F1 generation are partial result or the result which is intermediate of their parental genes. So now we are going to write the answer 13. First we are going to talk about codominance. We are going to write about codominance. Codominance. What is codominance? If a plant having yellow colored flowers and fuses with a plant having red colored flowers, then all the progeny of F1 generation will have orange colored flowers, If a plant having yellow colored flowers fuses or fertilizes with a plant having red colored flowers, what we are going to see in the F1 generation, all the progeny will have orange colored flowers. This is because both the alleles are dominant. Both the alleles are dominant and expressing themselves as a mixture. So both the alleles are dominant and expressing themselves as a mixture because, so this is a codominance. This is codominance. If we talk about the example, then example of codominance is human blood group AB. Human blood group AB is the example of codominance. If we talk about the next part, that is incomplete dominance, then incomplete dominance. In this, the genes of allelomorphic pairs are not expressed as dominant or recessive they expresses partially and forms an intermediate result in a hybrid of F1 generation. Now, if we talk about the example, a famous example of incomplete dominance is given in your NCERT, that is snapdragon flower. In the parent generation, the parents are having white colored flower and red colored flowers. When they produces their progeny, the F1 progeny, all the progeny in F1 generation are having pink color flowers, which is intermediate result of their parental gene. So this is the complete solution of question number 13, and this is the basic difference or explanation of the following terms such as codominance and incomplete dominance along with their examples. We have human blood group AB as example of codominance and snapdragon flower, which is showing pink color flowers in the F1 generation. All the progeny showing pink color flowers in the F1 generation is the example of incomplete dominance, where the intermediate result is coming out of their parental gene. So this is a complete solution of question number 13 over here. Now let's talk about the another question on the screen, that is question number 14. What is point mutation? Also give one example. So first of all, we have to understand what is mutation. So mutation is interchange, deletion or insertion. Interchange, insertion or deletion in a nucleotide basis of a DNA is known as mutation. When we're talking about point mutation, then this point mutation is a mutation when mutation is happening in only single nucleotide base. When mutation is taking place only single nucleotide base of a segment of DNA, then this type of mutation is known as point mutation. Then this mutation is known as point mutation. This point mutation can be done by addition of a single nucleotide base. It can also occur with deletion, with addition, with interchange of a single nucleotide. Single nucleotide when is inserted, deleted or interchanged in a segment of DNA, that mutation is known as point mutation. Alright, and if we talk about example, then for example we have sickle cell anemia. Sickle cell anemia, our erythrocytes, our red blood cells are having a gene, a normal gene, which is known as HBB gene. HBB gene have so many nucleotide bases. On the 17th nucleotide base, we have GAG. We have a codon named as GAG, which codes for amino acid known as glutamic acid. It is interchanged. The GAG, the codon GAG, the A between G and G, the A is interchanged by T. The A is interchanged by T. And this is causing a codon, this is changing a codon into GTG, which is coding now, which is making amino acid as valine. And this amino acid valine is producing sickle cell anemia in an individual. So now let's write answer of question number 14. What is point mutation? Before going on to point mutation, we will write a simple basic definition for mutation. So, interchange, deletion and addition in the nucleotide in the nucleotide basis of a segment of DNA, then this phenomena is known as mutation. This phenomena is known as mutation. When we are talking about point mutation, these things, interchange, deletion or addition, is going to happen only in single nucleotide base of a segment of DNA. So the mutation, we know now that what is mutation. So now mutation of single nucleotide, mutation of a single nucleotide base of a segment of DNA is known as point mutation. Is known as point mutation. All right. So if we're talking about example, we have sickle cell anemia as an example. Sickle cell anemia. What happens in sickle cell anemia? Suppose we have HBB gene, which is a normal gene for our erythrocytes, for our red blood cells. We have a normal gene named as HBB gene. HBB gene have so many nucleotide base pairs. So on the 17th nucleotide, we have a codon. On the 17th nucleotide, we have a codon GAG. In a normal condition, we have a codon named as GAG, which is coding or producing amino acid known as glutamic acid, which is a normal. And when point mutation occurs in any individual, what happens? This simple normal codon having GAG, this A is replaced by T and making it this codon GTG. A is replaced by T, making the codon GTG. Now point mutation occurred. The interchange of one nucleotide base has happened. The interchange of one nucleotide base has happened in a segment of DNA. A is replaced by T. So this codon is now going to produce another type of amino acid, which is known as valine, which is an abnormal condition. And this condition, when reaches into an individual, this produces the erythrocytes in a sickle shape, and this disorder is known as sickle cell anemia. So this is all about question number 14, and this is the complete solution of it. Let's talk about next question, that is question number 15, which is the second last question of this chapter. Who had proposed the chromosomal theory of inheritance? So this is a very interesting question, an important question, that the chromosomal theory of inheritance was proposed by two scientists named as Sutton and Boveri, but they were not able to prove their theory. They didn't prove their chromosomal theory of inheritance. This theory was proved by another scientist named as T.H. Morgan. So first of all, who had proposed the chromosomal theory of inheritance? The chromosomal theory of inheritance was proposed by two scientists named as Sutton and Boveri in 1902. They proposed this theory. According to this theory, they stated that the inheritance of chromosome in this theory, according to this theory, they stated that the inheritance of chromosome followed the same pattern of inheritance of genes as per the Mendelian laws of inheritance. So if we write answer 15, first of all, the chromosomal theory of inheritance inheritance, the chromosomal theory of inheritance was proposed by Sutton. There were two scientists, Sutton and Boveri. Sutton and Boveri are the two scientists who proposed the chromosomal theory of inheritance in the year of 1902, but they were not able to prove their theory of chromosomal inheritance. It is proved by another scientist named as T.H. Morgan. They were not able to prove their theory. According to this theory, or I may say this theory stated that the inheritance of chromosomes followed the same pattern, of inheritance of genes, followed the same pattern of inheritance of genes as per the Mendelian law of inheritance. As per the Mendelian law of inheritance. So who proposed the chromosomal theory of inheritance? There were two scientists named as Sutton and Boveri. They both together proposed the theory of chromosomal inheritance and in the year of 1902, but they were not able to prove this theory, and it is proved by another scientist named as T.H. Morgan. And according to their theory, they stated that the inheritance of chromosome followed the same pattern of inheritance of gene according to the Mendelian law of inheritance. So this is the complete solution of question number 15. Now we are moving towards the last question of this chapter, question number 16. So now let's moving on to the last question of this chapter, that is mention any two autosomal genetic disorders along with their symptoms. So we have to mention here two autosomal genetic disorders and their symptoms. So we are talking about two autosomal genetic disorders. First we are talking about or discussing about Down syndrome. Down syndrome is a genetic disorder which is autosomal, and this Down syndrome is autosomal aneuploidy. Autosomal aneuploidy, which is done by the extra copy, by gaining extra copy of chromosome number 21. If we are talking about the symptoms of Down syndrome, then the person who is having Down syndrome will have short stature along with small round head, furrowed tongue. Also the palm of that person is broad along with the characteristic palm crease. Physical, mental and psychomotor development is also retarded in the persons who are having Down syndrome. Now, if we talk about the next disease or disorder, that is phenylketonuria. Phenylketonuria is also an autosomal recessive disorder which occurs because of the lack of an enzyme which converts phenylalanine into tyrosine. The person who is having phenylketonuria shows mental retardation because of the accumulation of phenylalanine. Phenylalanine in that person is get converted into phenylpyruvic acid and their derivatives. This is the condition of phenylketonuria, and this the person who is having phenylketonuria is actually shows less absorption by their kidneys or very poor absorption by their kidneys. Alright, so if we want to write the answer of question number 16, first is Down syndrome. We are talking about Down syndrome. So it is a case of autosomal, it is a case of autosomal aneuploidy. It is a case of autosomal aneuploidy and arises because of gain of extra copy of chromosome number 21. Now if we talk about the symptoms of Down syndrome, then the person who is having Down syndrome, the person is short statured, along with small round head, furrowed tongue. The palm of that person, is broad with characteristic palm crease, Alright, the palm of a person who is exhibiting or having Down syndrome disorder, the person's palm is broad and also having characteristic palm crease. Also physical, psychomotor, and mental development is retarded in these persons. The next genetic disorder, which is autosomal, we are talking about is phenylketonuria. This is second and this is one. Phenylketonuria. Now what happens in this? First of all, this is autosomal recessive disorder. This is autosomal recessive disorder which happens because of lack of an enzyme which converts the phenylalanine into tyrosine. So the person having phenylketonuria shows mental retardation because of accumulation of phenylalanine that converts into phenylpyruvic acid which goes out or throws out in urine. Alright, so this is all about the question number 16. This is all about the two genetic disorders which are autosomal, that is Down syndrome along with its symptoms, and the another type of genetic disorder which is autosomal along with its symptoms is phenylketonuria. Now with this we are ending this session, and we already have completed all the NCERT questions given in the NCERT, that is question number 16, and this is all for today. Thank you and have a nice day.